Good morning, good afternoon, and good evening, everyone. And welcome to this Inside Scientific webinar titled Molecule Transport Across Cell Membranes, Electrochemical Quantification at the Microscale. I'm Liam Sanyo from the events team here at scientist.com, and I'll be your host today. This webinar has been sponsored by Harvard Bioscience and its affiliate ECA, so a big thanks to them for making this webinar possible. We're very fortunate to be joined by Dr. Sabine Kuss, who's Associate Head of Graduate Studies and Associate Professor in the Department of Chemistry at the University of Manitoba, who will discuss the importance of transmembrane molecule exchange and how you can detect and quantify membrane transport of molecules in cells using electrochemical and electrophysiological methods. And with that, I'm very pleased to welcome Dr. Sabine Kuss. So Sabine, thanks so much for joining us today, and the floor is yours whenever you're ready.
Yeah, thank you, Liam, for the kind introduction. And I'm excited to be here today and talk about molecule transport across cell membranes and how we can use electrochemistry to not just detect these events, but also to quantify them. All right, so I want to start by acknowledging the people behind the experimental work that I'm going to share here today. So my PhD students, specifically Luma Clarindo Lopez and Nikita Thomas, and undergraduate students Shubneet Thind and Roy Dao. So funding agencies who carry our work are mainly NSERC, Research Manitoba, and Canada Foundation for Innovation that supported the purchase of the technology that I'm presenting here today. So before we dive into electrochemistry, let me touch upon the basics of transmembrane transport events. So what we are looking at here in the top left image is the structure of a cellular membrane, as you might know it. Yeah, so we have a lipid bilayer consisting of hydrophilic heads and hydrophobic tails containing cholesterol, sugars, and proteins, which can be involved in molecule transport. So in tissue cells and bacteria, substances are transported through three main biological entities, shown here in the lower right image. So lipid bilayers, porins, and active pumps. So lipid bilayers allow the simple diffusion of molecules across membranes. Porins facilitate this diffusion. And efflux pumps are proteins that are designed for the export of potentially harmful substances from living cells. So efflux pumps allow for active transport. They require energy and move particles against a concentration gradient. All right, and a specific cellular characteristic that depends on molecule transport is drug resistance. So you might have heard about this phenomena, as it is an issue in every country on Earth, and it enables cells to withstand antibiotic treatment or chemotherapy. So statistics shows that 10 million deaths worldwide each year are attributed to antibiotic resistance, and the statistics for cancer resistance for chemo resistance is very similar. So therefore, various national and international health organizations, such as the United Nations, the Public Health Agency of Canada, have called for an urgent development of new treatment and diagnostic strategies. So examples for drug resistance mechanisms are shown here in the schematic for bacteria and mammalian cancer cells. So we are looking at membrane-related mechanisms, such as membrane protein modifications that prevent an organism to consume a certain medication. Then we have efflux pump activation, which expels medication rapidly before these drugs can take action. So then these efflux pumps are similarly expressed in mammalian cancer cells, shown here on the right, where a drug can be either expelled directly or in conjugation with glutathione. There are also intracellular mechanisms, such as drug target alteration, where the drug cannot bind to its intended target, or the drug inactivation by enzymes. And today I'm going to discuss how we can use electrochemistry to detect any of these mechanisms. All right, so there are many bioanalytical methods that aim to quantify the transport of molecules from cells, such as mass spectrometry or chromatography methods. And in our recent publications at Angewandte Chemie, we summarize these methods and show these approaches for the accumulation methods and dynamic methods. So if this is a topic that interests you, I would like to refer you to this publication. It's also provided as a handout here to this webinar. So most of these approaches are accumulation methods, these non-electrochemical approaches. These are quantitative, but often lack sensitivity or cannot analyze complex samples, such as a cell. So in any case, they are unable to measure events as they happen, which results in the loss of analyte during the experimental procedures. And today we will talk about dynamic methods in electrochemistry, which will quantify molecules immediately as they emerge from cells. Okay, and now for those of you who are not well versed with electrochemistry, here is a short one-on-one. So before we can measure complex biological events, such as the transport across cell membranes, we need to understand the electrochemical signature of the molecules of interest, which could be pharmaceuticals that cells consume or expel. It could be metabolites that we use as indicators for diseases or for the general redox state of cells. Or it could be a redox mediator that interacts with molecules that are being emitted from cells. In any case, electroanalytical chemistry involves an electrochemical cell that's shown here on the right, where we have a vial that contains an electrolyte. So in terms, when we measure cells, this is an aqueous solution, and we have 3 electrodes. Our working electrodes is where the chemistry happens. So this is mainly a metal wire, and it's sealed in a glass capillary or any insulating material. It could be epoxy as well. Then we have an auxiliary electrode that closes the circuit, and we have a reference electrode that gives us a steady reference point so that other people can understand the voltages and potentials that we are measuring across the world. So here you see on the left-hand side an scheme of such a working electrode. So the yellow part is the metal, and we can apply a voltage to this metal and drive an oxidation reaction. So here we show the diffusion of a redox active species in solution towards this electrode where it gets oxidized. And these redox reactions involve then electron transfer reactions. So we are basically counting electrons and can thereby quantify the reactions that happen at the electrode tip. So when we plot the data that we receive from this, we are plotting it as a current potential plot oftentimes, that resembles when we sweep the voltage forward and backwards a duck, and it's so the shape of a duck, if the reaction is chemically reversible. So here in this figure, you see in red an oxidation peak and a reduction peak in blue if the reaction is chemically reversible. And this data can actually provide us with a lot of information about our biomarker or indicator of interest, our analyte. So we can read out oxidation and reduction potentials. We can investigate the diffusion behavior of these molecules in solution and how they interact with our electrode. We can study pH dependence, we can determine detection limits, and we can also look into interferences of ions or competitive analytes, such as drugs. So here on the right-hand side, you see examples of these voltammograms for different antibiotic species. So in blue, you see the electrochemical signature of tobramycin, a very common antibiotic, as well as in green, ciprofloxacin, and in red, we see an antibiotic hybrid. So You can see that these reactions are not chemically reversible because we don't see a reduction peak, but we do see a pronounced oxidation peak for both tobramycin and ciprofloxacin. When we investigate hybrid molecules that are basically designed to trick cells in consuming a certain antibiotic they might be resistant to, we see in the red curve that there is a pronounced oxidation peak that corresponds to tobramycin, but also a small shoulder peak that is attributed to ciprofloxacin. Yeah? And then we can, in electrochemistry, we can define our parameters such as how we apply the voltage in terms of duration, in step height, and step width, and which is called differential pulse voltammetry, to get a better resolution of these peaks. And these peaks we can then use to quantify an analytical species in solution. Yeah? So here in this figure, you see in figure B how we use this to determining limits of quantification and limits of detection for a certain species. And we can carry out these measurements at different pHs, as shown in D, where we then assess how stable these measurements are across different pH values. Okay, and with this small introduction, I will jump right into some of our results where we are detecting membrane-related transport mechanisms. Yeah, so here we are aiming to detect drug susceptibility in Pseudomonas aeruginosa bacteria because of their clinical relevance. Yeah, so these Pseudomonas bacteria are involved in acute infections, but also in chronic diseases such as cystic fibrosis. So here you see a very simple electroanalytical approach to quantify drug susceptibility. So it's based here on a solution of bacteria that we expose to an antibiotic. Here in this case, it's tobramycin. And then we give the bacteria time to uptake this drug, and then we centrifuge the solution to separate the supernatant and the bacteria. So our hypothesis is that if bacteria are resistant to tobramycin, they would either not consume this drug or expel it after the consumption. So basically leading the drug back into solution, and we would obtain a higher electrochemical signal than in susceptible bacteria, which would consume and retain this drug and would be removed with the drug together from the solution. Okay, and here you see a representative experimental plot of this experiment, so carried out in differential pulse voltammetry. So here in black and red, you see our controls. Yeah? So control in black represents a known concentration of tobramycin in our vial before we expose it to bacteria. And in red, it is the same vial after 15 minutes of incubation, and you see there's no degradation in concentration of these molecules. So then we expose the solution to different bacteria strains. In green, you see a susceptible bacteria strain, yeah? And you see that the signal is much lower. This is because these bacteria are able to consume tobramycin in solution and have then removed it together with the organisms through the centrifugation. We have a resistant strain in pink. Yeah, so these are bacteria where we know that they are resistant to tobramycin because of efflux pump expression. And you see that the signal is much higher because these organisms are able to feed back this drug into solution. And in blue, we have a mucoid strain of these bacteria, which are even more resistant because they express certain features and fragments that can render them even more resistant. Of course, we don't carry out these studies just once, but we are doing a large analytical investigation here. So the plot that you have just seen here is one experiment that corresponds to the highlighted section here for 15 minutes of incubation. But we also investigated longer incubation times where we see that this difference in current response is not as pronounced. And we validated this with fluorescent microscopy because these cells are just too stressed. They are breaking apart, and tobramycin is given back into the solution before centrifugation if the incubation time is too long. Yeah. So we see here that 15 minutes of incubation is very suitable to not just detect a drug susceptibility by electrochemistry, but also to differentiate not only between susceptible and resistant organisms, but also between different degrees of resistance. All right, but what if it is not an efflux-dependent mechanism, yeah? So to investigate these intracellular resistant mechanisms, you use the technique of scanning electrochemical microscopy. So SECM is actually an incredible tool that lets us track how many molecules interact with a sample, such as a living cell per second. All right, so scanning electrochemical microscopy is a technique that was invented a couple of decades ago, but has increasingly received attention for the biological in the biological field over the last decade. So here we see the heart of this instrument is a microelectrode shown in the right-hand side figure, which is again a metal wire, but this time on the micrometer scale, that's sealed in a glass capillary. Yeah? And you see here, when we look at the tip, we see this metal in the center and the glass surrounding this metal where we carry out the electrochemistry. Yeah? So this electrode is mounted onto an Z-axis positioner that's shown in the left-hand side figure in orange. And this Z-axis positioner can approach the sample precisely in 1 nanometer steps. And we can position this electrode very precisely at a given height or desired height on top of a sample. So our sample actually sits in a Petri dish, and that is mounted onto an XY axis positioning stage. So that moves in X and Y. So we have all 3 dimensions to position our electrode in close proximity to a biological sample. We also have an optical microscope and a fluorescence module integrated. So that lets us observe the sample at the same time as we conduct our electrochemical measurements. We can stain different cell types, different cell strains, different cell components by fluorescence so that we are aware of what we are measuring and where we are with our electrode, or to identify a cell if we want to re-image them. Yeah? The electrochemistry is controlled by a potentiostat. Okay, so now I'm going to show you how we investigated chemoresistance mechanisms by SECM. So we are looking here at intracellular mechanisms. We have done similar studies that I've just shown for bacteria with these cancer cells, where we did not get a significant difference between these cells that we know are resistant to a certain chemotherapeutic or susceptible. Yeah. So that led us think about, so what can we do to investigate intracellular mechanisms? And one molecule that comes to mind is glutathione. Glutathione is one of the major antioxidants inside cells, which controls the redox state of the cells and is directly involved in the efflux or the handling of harmful molecules inside cells. So in the past, we have shown, our literature shows, that we can use a redox mediator called ferricin methanol, shown here in green, to track the concentration of glutathione that is exported from cells. So ferricin methanol is oxidized at our microelectrode that is positioned in close proximity to our cells. And it's also passively diffusing inside of cells. And when it does so, it increases the ratio of reduced glutathione and oxidized glutathione to enhance the efflux of reduced glutathione from the cells. And then when it does so, when the electrode scans over our cells, it is basically regenerating the ferricin methanol by the oxidation of the glutathione. Yeah, that is shown here in the top left scheme. And as a result, we get these beautiful diffusion maps of cells and the surrounding glutathione. So in this, in the figure below, you see in the blue is our Petri dish, and you see as green and these colors basically how the glutathione is diffusing from cells and creates the sphere around them. Yeah, so that is a very good visualization of the events that we are tracking here. Yeah, and we do this for ovarian cancer cells that are resistant to a chemotherapeutic called carboplatin here. So carboplatin is one of the most widely administered chemotherapeutics across different types of cancers. And we have cells on the right-hand side that are susceptible to carboplatin. But how can we now quantify this, the resistance phenotype of these cells? So when we scan over the cells, which is shown here, indicated by this arrow, we can carry out these line scans at different scan rates. Yeah, and when we do so, we get an increase in electrochemical current that's shown here on the right with increasing scan velocity. This is because when our electrode crosses across a cell, we basically squeeze the fluid in between the cell and the electrode, and the faster we do so, the higher the accumulation of electroactive species is in between the electrode and the cell. I will come back to that point in a minute. But what we get is we obtain a profile of line scans that increases its current with increasing velocity. And then we can select these peak currents, so the highest point of these line scans, and plot them as a function of scan velocity. Yeah? So on the left-hand side, you see the carboplatin-resistant cells and basically presenting the peak current as a function of scan velocity, and we obtain a slope. Yeah? So now let's have a look first at the black curve. So you find the black line, which is right in the center there, and this is our initial slope. So these are the cells before we treat them with carboplatin. And now we are exposing these cells and carry out these line scans every 5 minutes. Yeah? And you see the very first line scan that we do immediately after exposure represents a change of slope. Yeah? So the slope increases before it then relaxes back. Yeah? And it's very stable over time, over a time period of 30 minutes. When we look at the susceptible cells on the right-hand side, however, you see that after exposure, this slope gradually decreases. Yeah? So this is already like a qualitative appreciation of the differential behavior between resistant and susceptible cells by electrochemistry. So we can then quantify the current. Yeah, so when we have our line scans, we can integrate underneath the curve. We can calculate the numbers of molecules that are being converted at the electrode, and we can plot the current percentage that it is represented by these line scans. Yeah, so here you show one example for a scan rate of 100 micrometers per second across these cells, and we see our initial current value there set at 100%. And now we look at our control cells, the susceptible and resistant cells that do not significantly differ without exposing them to carboplatin, because their intrinsic glutathione ratio seems to be very similar. However, when we expose them to carboplatin, we see that the current values for the resistant cells significantly increases with a confidence interval of 99%. And in the susceptible cells, it decreases. Yeah? And this is one of the very first quantifications of these responses to the exposure of a chemotherapeutic. Okay, so now we have talked a lot about drug resistance and using electrochemistry to detect this phenomenon. Now I want to shift gears a little bit and show you how we can also use SECM to validate biomarkers for disease detection. Yeah? And for this purpose, we are looking at the impact of cytochrome C oxidase deficiency. So cytochrome C oxidase is a large transmembrane protein complex. It is the last enzyme in the respiratory electron transport chain. There it receives electrons and transfers them onto oxygen so that the ATP synthase can synthesize ATP. So, and of course, like respiratory chain, you can imagine in how many organs this enzyme is involved. Yeah, so if there's a deficiency due to the absence or abnormality of the protein cytochrome C oxidase, this leads to severe muscle weakness, heart and liver, kidney problems, and even brain damage. So this is a disease that affects newborn babies. And right at the moment, there is no routine screening for this deficiency until symptoms present themselves. Yeah. However, if this disease is caught very early, there is medication that can be administered to minimize these effects on the human body. Yeah. So we are investigating the possibility of having an electrochemical sensor developed for the routine screening of this disease in newborn babies. So in this research, we are looking at fibroblast cells that are extracted from connective tissue in muscles, where we want to establish a proof of concept. Yeah, we want to demonstrate that we can use electrochemistry to quantify cytochrome C oxidase deficiency in these cells. Yeah, we did some Western blot studies here to show that these cells indeed are lacking the expression of the SCO1 metallochaperone protein that is required for the proper assembly of the COX protein. So yes, so we are using here a control strain and a patient strain called SCO1 that was extracted from patients. Yeah, so the VDAC here in this Western plot is just our control protein, and then in B you see the assembly of this complex 4 that is incorrect in these patient samples. Okay, so our redox mediator here is tetramethylphenyldiamine. Yeah, and so the connection here is that tetramethylphenyldiamine, or short TMPD, has been used for many years in cell culture to evaluate the presence of cytochrome C oxidase in bacteria. So this is actually a transparent solution that is being dropped onto bacteria on an agar plate. And if these bacteria are cytochrome C oxidase active, they have the ability to oxidize TMPD to its radical cation, which has a very characteristic blue color. And we aim to transfer this principle onto electrochemistry, which we think is more sensitive and can detect lower levels of cytochrome C oxidase expression. So in our studies, we are using fibroblast cells, and here in this left image, you see how our electrode scans across these cells in our SECM imaging. Yeah, and when we apply a reduction potential, the hypothesis is that the cells are able to oxidize TMPD, where what is reduced at the electrode, and which gives us a feedback loop, and we should be able to obtain current values that correspond to the expression of cytochrome C oxidase activity. So we have here on the right-hand side a line scan across these cells. In black is just shown the Petri dish. And then we see in red, when we apply a reduction potential, we can detect the cytochrome C oxidase. And if we apply an oxidative potential, we can even image the cytochrome C reductase. So this scheme shows you a little bit what we are proposing is happening inside the cell. And let me guide you through this scheme here. So in scheme A, you see the conditions under normal aerobic conditions, yeah, where cytochrome C oxidase, shown in purple, catalyzes electron transfer from the reduced cytochrome C to molecular oxygen. Yeah, the cytochrome C oxidoreductase, or complex 3, it's also called, that's shown in orange, then catalyzes the electron transfer from ubiquinol to oxidized cytochrome C. So in our SECM studies shown in B, we scan then across single cells or groups of 2. And here the electron transfer can occur directly from TMPD to cytochrome C oxidase without the involvement of cytochrome C actually. So in scheme C, we propose that since TMPD is a membrane permeable redox mediator, it can easily penetrate a cell's bilayer membrane and diffuse to the mitochondria. So once oxidized, the TMPD radical cation passes then through the outer mitochondrial membrane and the plasma membrane of the cell to interact with the surface of our microelectrode. And this gives us this feedback loop that we are observing. So long story short, TMPD gets oxidized by the cells and reduced at the electrode. Okay, and our SECM 3D images show us these plots where we can already anticipate the results. So you see in A and B our control signals, and when you compare them to our patient samples in C and D, you see that the oxidation as well as the reduction is much more pronounced in our control strains. But how can we quantify this now further? So for this, we are carrying out our line scans at different velocities, as I explained earlier. And also here we can appreciate qualitatively already a difference. But we really want to extract a numerical value for this differential behavior of these cells. So for that, we carry out our line scans, and I already spoke to the fluid movement and the electrode position on top of the cells, which is illustrated here in figure B. So, and these tiny arrows that you see there indicate the movement of the solution in between the electrode and the cell, where we basically accumulate the electroactive species here in solution. So the beauty of the system is that the topography contribution that is coming from the body of the cell underneath this electrode so the contribution to the electrochemical signal varies, whereas the reactivity contribution, so the regeneration of our redox mediator remains constant. And because of this fact, we can decouple the topography and the reactivity by numerical modeling. We are working in collaboration with Dr. Dao Trin at the Université La Rochelle in France. And he is an expert in SECM, modeling for biological applications. And let me walk you a little bit through this model. So in this model, there are 2 ways to extract a numerical value for the substrate kinetics. So here we are calling the substrate kinetics the apparent heterogeneous rate constant. So we can do this in 2 different ways: at high scan rate, high scan speed SECM, or at regular speed SECM. So high scan rate means anything above 100 micrometers per second. So we like to normalize the velocity in a way you see here in this equation, where PS equals the V is the velocity, A is the radius of the active area of the microelectrode, D is the diffusion coefficient, and the small d is the tip to substrate distance. So in this graph, the curve represents our experimental data. So if you're able to scan at high scan speed SECM, we are looking at the region of the graph that is linear. So the function in this case that is fitted to the experimental data is shown here, and on the top in green, and depends on the slope of the curve mainly. In the second case, so if you're using regular, like us, SECM scan rates, you have to fit a more complex function that gives not 1, but 3 fitting parameters, the variables I0, R0, and A. And these are numerically determined for acquired experimental data and are depending on this kinetic K that we are looking for. And what you obtain is basically a plot that shows the kinetic rates ranging from fast kinetics to that were established at a mercury drop in literature, and to slow kinetics. And as you can see, our cells here lie in between, and we can extract a apparent heterogeneous rate constant K0. So let's come back to our cytochrome C oxidase active cells. So our control cells here exhibit a kinetics of 3.2 times 10 to the power of -5 meters per second. And you can see that our patient cells have a reduced cytochrome C oxidase activity by almost half. We can also analyze the cytochrome C reductase. Although this has not been altered in these organisms, it is still, we can see a trend that also here the reductase is slightly decreased. Great. So now we have talked about metabolites. We have talked about redox mediators that can be used as indicators for diseases. And now in this last 5 to 7 minutes, I would like to talk about how we can monitor or aim to monitor reactive molecules that are transported across membranes. So, and for this purpose, we are using optical fibers that we are integrating into our microelectrodes. So if you remember, our microelectrode so far contains just a metal wire as a disc surrounded by glass. And now we're gonna integrate an optical fiber into this electrode as well. And this is not the first time people have been doing this. We were not the first. In literature, you can find various very interesting approaches where the glass is either a ring around a metal, shown in A, or we have a side illumination or detection of light-sensitive events at electrodes. But as you can also appreciate on the right-hand side, the geometry of these probes in the past has not been very consistent. And especially when we image very complex systems like cells, the reproducibility and repeatability of experiments is incredibly important because we need to gather a large amount of analytical data sets. So we were aiming first to develop a method for the development of these micro-optical ring electrodes for SECM applications of biological samples. So the fabrication procedure is shown here on the right. That starts with a glass capillary in step 1 that is filled with epoxy. And then we use an optical fiber that is gold-coated and push it through this partially cured epoxy. And then once it's fully cured, we can polish the tip in step 3 and connect the copper wire to assemble a micro-optical ring electrode that resembles very much the dimensions and features of a regular ultra-micro electrode that we have been using in the past. All right, so the tips of these micro-optical ring electrodes can have various shapes. Here in the upper left image, you see just the gold-coated optical fiber that we then integrate into our electrodes and into our capillaries. And you see that we can produce conical shapes. We can produce rods at different sizes in diameter and position them then. The aim is to position them in close proximity to a biological membrane that's shown in the lower right-hand side figure and perform SPECM cell irradiation and imaging. So here we aim to irradiate a sample through the optical fiber and trigger light-sensitive responses from the cells and record the electrochemical response simultaneously. So to this end, we integrate our micro-optical ring electrode, short our MORS, into our SECM instrument to build an SPECM, a scanning photoelectrochemical microscope. Yeah? So you see here on the left-hand side, what has changed now is that we have a spectrophotometer attached to our MORE to irradiate a sample and to do, concurrent electrochemical measurements, or we can use bottom illumination through our microscope and then carry out spectroscopy through the optical fiber as well. So this is our instrument here from Harvard Bioscience and HECA. Yeah, and I want to acknowledge these people here, especially Frank, who has been incredibly supportive and is guiding our research in a tremendous way. And I just hope, guys, just keep doing what you're doing, because this system is just amazing. And we have, here the proof of concept for carrying out spectroscopy through these optical fibers. Yeah, so we're using here again our TMPD because it has this characteristic blue color, and you can compare the optical fiber spectrum collected through the optical fiber on the left, and you can compare it to a spectrum that was acquired through our MORE. Yeah, so this is still under investigation, and we are now hoping to being able to identify molecules that contribute to the electrochemical current signal by spectroscopy, by combining spectroscopy with electrochemistry. Okay, so now our biological sample here are algae cells. Yeah, we first had to prove that they are indeed light sensitive. So we exposed them here for 3 hours to light or 3 hours in darkness. And when we carry out voltammetry in a beaker containing these cells, we can appreciate that these cells under light condition generate oxygen. Yeah. And whereas in dark, we get a much more reduced current signal here. Yeah. So then in our SPECM stimulation experiment, we are scanning across single cells of algae and record the current while either switching the light on or off at our electrode. Yeah. So first have a look at the red curve here, where you see a decrease in current when the electrode scans over these cells. Yeah. And this is a big contrast to when we switch the light on and we perform these scans across cells, where you see a pronounced oxidation peak, the oxidation of oxygen that is basically produced at the electrode. I have to correct myself here a little bit because we are performing oxygen reduction at the electrode, but the cells are producing oxygen. Yeah? And yes, so we can also quantify this current that is shown there in the inset, which is statistically significant. All right, so where do we go from here with this technology? So future potential applications, we want to focus our attention to reactive oxygen species and reactive nitrogen species that are emitted from cells, and then ideally use the optical fiber to also use spectroscopy to differentiate between different ROS and RNS species that might be involved in cancer progression or cancer initiation studies. Okay, and this brings me to the summary here. So we talked a lot about SECM and SPECM. We talked about the detection of molecule transport by voltammetry. I hope I convinced you that we can quantify membrane-related drug resistance mechanisms by voltammetry and monitor the quantification of metabolite export, such as glutathione and oxygen from cells. Yeah? And we also discussed the applicability of SECM to detect diseases at the example of cytochrome C oxidase deficiency. And with that, I thank you very much for your attention. If you're interested in what we are doing and would like to discuss more outside of this webinar, please get in contact. You can find us online or on Twitter, or it's now called X, I believe. Yeah. So thank you again for your attention. And I hand it back to Liam, I believe.
Excellent. Well, thank you so much, Sabine, for a fantastic presentation. And as a reminder, if you do have a question that you'd like to ask, navigate over to the Q&A tab and submit your questions. It'd be great to hear from you. So let's kick things off with a nice question here Sabine, what sort of expertise in your lab do you need to perform SECM experiments on biological samples?
Oh, wow. I mean, it is really, we get students who come from various backgrounds. So we have students that are coming from biology and have never done any electrochemistry. And that is not a problem at all. We can train anyone really who is very, who's passionate and generally interested in learning a new method and technique. This is how I started, right? I started out with an undergraduate degree in biology. We have also students coming from biotechnology. So they have a little bit of engineering experience, I would say. So for them, it's easier to, actually build these electrodes, and they are very interested. So it depends what people are interested in. But if people are coming from oncology, for example, and want to study these related mechanisms of cancer, we would be happy, to engage and collaborate, or to, train people new. But I don't think there is a specific expertise you need to know in order to get familiar with the technique. But yeah, it depends what the application would be. Mm-hmm.
Fantastic. Yeah, that's great to hear, especially considering when we look back at the poll results, where we saw that there's people from a pretty wide range of disciplines
Oh, yeah.
here.
Mm-hmm.
All right. So next question here. So what sorts of considerations do you need to have when using biological samples in this imaging environment?
Yeah. Oh, that's a very good question. So of course, we didn't have the time here today to talk about this in detail, but imaging cells is a very complex endeavor. Yeah. So we image different types of cells, not just bacteria and mammalian cells, but cells have individual requirements, right? They grow at different temperatures, for example. And temperature control is an incredible factor that needs to be considered. We have another publication currently under review that actually shows that minor temperature changes of about 1 degree will alter the kinetics of the cells. Yeah. So although cells seem stable, and they are not dying because, you're just pre-warming your media and then you image them over, like, let's say half an hour, and they don't die, doesn't mean that the results that you collect are accurate, right? So in our team, we really pay close attention to these experimental factors, temperature, but also light exposure. Yeah, the composition of your medium, your cell medium they grow in, and that we use as an electrolyte in electrochemical studies. So there are, various parameters that need to be considered. Thank you for this question.
Fantastic. Well, thank you. Another question here. So in your study of chemoresistance imaging, it seemed the target cell showed two hotspots with higher tip currents. And could you please explain a bit more about this in terms of why two hotspots and not just one were observed?
Okay, maybe I can go back to the slide. Liam, do you have an overview of the slide and can guide us right to
Yeah. If you click Slide Navigator, it should be able to...
Oh, yes, I see. Okay. Okay, are we talking about this one? So these are actually two cells that are sitting side by side. So we are actually trying not to image. So when we expose cells and we grow them in our Petri dish, we have to do so over, let's say, a duration of 6 hours until they settle and they are fully established and their metabolism is at an equilibrium before we can image them. During this incubation or pre-incubation period, these cells often divide. Yeah. And depending on the cell line you're working with, these cells might divide within 2 hours or 12 hours. Yeah. So when we look at these ovarian cancer cells here that divide, I would say every 6 hours, we do not want to look for a single cell, because after like 6 or 12 hours, if you see a single cell, something might be, not totally accurate or, correct with it, right? So we are looking for a group of two. Yeah, and then we scan here across one of these cells. Yeah, so there is a discussion about how to carry out line scans across two cells that are sitting side by side. Yeah, because you can basically scan it as it is shown here with this arrow just across one cell, or you can come from the other direction and scan across both cells. Yeah, the signal of one cell will influence the other. But because they are sister cells, and as long as you're consistent in the methodology that you're applying here to image, we believe we can trust the data, because we are, if one cell influences the other, it does so for every experimental data set that we are collecting. Yeah, again, that's a good parameter that needs to be focused. I hope that answers the question.
Yeah, I think so. Excellent. Thanks a lot. All right. So, Sabine, how can we work SECM over biomaterials coated on flexible substrates?
Great question. Biomaterials coated on flexible substrates. So SECM has been used in the past to basically characterize, electrochemical surface properties. Yeah. So when I hear flexible substrates, for me that basically comes with with a lot of variation in height of the surface. Yeah, soft surfaces are not as easy to image. There are approaches. So the data that I've shown here today is carried out in constant height imaging, where we keep the electrode at a certain height and we scan across our cells. This is possible because our samples sit on a petri dish that's fairly flat. Yeah. When we come to soft surfaces, this variation in height of the substrate is much larger. So constant height imaging might not be, the best approach. But there are shear force approaches where the electrode basically records a shear force signal that is based on the amplitude or phase of a vibrating tip, and then can track the surface features very precisely. Yeah. So when the amplitude or phase changes in about 200 nanometers across the surface, when the surface topography changes, the tip retracts to keep a certain signal. So that's shear force, SECM approaches to keep a constant distance. I think this would be more appropriate for soft surfaces. Yeah, but it is not trivial to conduct. Yeah.
Excellent. Great answer. Would the shape of the probe alter the resolution of the peaks, do you think?
The shape of the probe, yes, absolutely. So the diameter of the electrode that we choose has a purpose. Yeah. So here, when we scan large biological cells, we are choosing a size of electrode that resembles about the same diameter of our cells, because we want to collect all molecules that the cell is releasing. Yeah. So like a vacuum. Yeah. But if people would like to study higher resolution or features or features that are smaller than micron size, yeah, or tens of micrometers, then we can reduce the size of the electrode to obtain higher resolution images. Yeah, there are even nano electrodes that we can fabricate. Also in our group, where we pull a microelectrode to nanometer size dimensions. Yeah, and so it depends on what you want to image, but it is certainly possible to increase and play with the resolution. Mm-hmm.
Fantastic. All right, I think in the interest of time, we'll just have one last question. So maybe one looking forward would be great. Are there any research avenues or developments in the field that you're particularly interested or excited in pursuing moving forward?
Yes, definitely. So we are studying biological systems, both bacteria and mammalian cells, tissue cells. So we are still interested in drug resistance primarily, and we study this to unravel mechanisms, to understand mechanisms behind resistance. For example, the example I showed here today with carboplatin, the mechanism or mechanisms that are involved in this resistant phenotype are not fully understood. So we are trying to, on one hand, understand mechanisms in resistance, and also we are aiming to develop point-of-care devices, so tools like an electrochemical sensor that we can distribute to physicians, hospitals, in clinics, in remote areas to make better decisions about diagnostic and treatment of diseases. Yeah, let it be the administration of antibiotics, but also to detect diseases such as cytochrome C oxidase that I have shown here. Particularly interested I am to investigate biofilms in the future. So, and we have a nice idea I would love to talk about, but I'm just gonna keep it for myself. But if you're interested in biofilm analysis by electrochemistry, please get in contact, and we would be happy to talk more about it.
Fantastic. Yeah, super exciting field. All right. Well, Sabine, again, thanks so much for sharing your insights today. It's really been a pleasure talking with you today.
Thank you. No, thank you for having me.
Yeah, my pleasure. Also, a big thanks also to the audience for joining us. And in closing, we hope you enjoyed this Inside Scientific and Harvard Bioscience webinar, and we'll see you again next time. Have a great day, everyone.